We know that gene interactions occur when two or more allelic or non-allelic genes of the same genotype influence the outcome of particular phenotypic characters. Can you specify the two broad categories of gene interactions? Yes. They are interallelic gene interactions and non-allelic gene interactions. Do you remember that gene interactions cause the departure from the normal Mendelian phenotypic dihybrid ratio that is given as 9:3:3:1? This is true because we have seen that non-allelic interaction involves two or more independent genes present on the same or different chromosomes, interacting to produce a new expression. Now, let us look at one such type of non-allelic interaction, the epistasis. Epistasis is a phenomenon that takes place when the presence of one gene at one locus suppresses the action of another gene at a different locus. In this condition, the presence of recessive gene EE coding for yellow suppresses the effect of BB or BB coding for black and brown during epistasis. Here, when the genotype is BE, then the coat color is black. When it is BBE, the coat color is brown. When it is BEE, then it is yellow, due to the presence of EE. And when the genotype is BBEE, then the coat color is yellow, due to the presence of EE. The gene that suppresses the effect of another gene is called the epistatic gene, or inhibitory gene, while the gene whose effect is masked or suppressed is called the hypostatic gene. What is a Punnett square, and how can it make the cross a simple one? A Punnett square is the graphical representation of the probable genotypes as the outcomes of a particular cross. We can assess all the possible combinations in an offspring. Let us represent the epistasis with the help of a Punnett square by showing a cross between two white flowers. Here, a cross between two white flowers with genotype CCPP and CCPP resulted in the formation of a purple flower with a genotype CCPP in the F1 generation as a hybrid. Expression of purple color is possible, as both the dominant genes C and P, responsible for purple color, are present. In the next step, the selfing of the hybrid resulted in the formation of purple and white flowers in the phenotypic ratio of 9:7, respectively, during F2 generation. Here the deviation of F2 generation ratio from the normal dihybrid ratio 9:3:3:1 to 9:7 proves the epistatic interaction among the genes. Here, CC is epistatic to PP or PP, and PP is epistatic to CC or CC. Furthermore, epistasis is of different types: recessive epistasis, dominant epistasis, duplicate recessive epistasis, duplicate dominant epistasis. The first type of epistasis is recessive epistasis, also called supplementary epistasis. This takes place when recessive alleles at one locus mask the expression of both dominant and recessive alleles at another locus. BB is dominant representing black, while BB is recessive representing brown. The expression of black and brown colors are masked by the presence of EE. Here, EE is epistatic to BB or BB. The second type of epistasis is the dominant epistasis, which occurs when the expression of both alleles, dominant and recessive, at another locus, is masked by a dominant allele at one location. The white color is controlled by W, and the yellow color by G. White color is dominant over both yellow and green. The green fruits are formed in recessive conditions of both genes. Here, WW is epistatic to GG and GG. The third type of epistasis is the duplicate recessive epistasis, also called complementary epistasis. This occurs when recessive alleles at either of the two loci can mask the expression of dominant alleles at the two loci. Here, PP masks the dominant CC, and CC masks the effect of PP. These are epistatic genes. Hence, purple is not expressed when both alleles of a recessive gene are present. The fourth type of epistasis is the duplicate dominant epistasis, also called duplicate gene action. This occurs when a dominant allele at one of the two loci can hide the expression of recessive alleles at both loci. Here, AA and BB are dominant and can mask the effect of AA and BB. Hence, AA and BB are duplicate epistatic genes. Hence, when both AA and BB are absent, then AA and BB express themselves as recessive genes. Therefore, we have understood the concept of epistasis and its types.